Greetings, everyone, and welcome to the Science Fair on Reshaping Science and Health by collaborating between borders and disciplines. I'm Judy Sakanari. I'm a professor in the Department of Pharmaceutical Chemistry at the University of California at San Francisco. Today, we will have a great panel discussion on global issues of parasitic diseases and collaborations across the borders. Before we begin our session, I just want to give a couple acknowledgements. We would like to thank Nevin Krogan and Jacqueline Fabius of the Quantitative Biosciences Institute, or QBI, here at UCSF, as well as Frederick Jung, who is the Consul General of France in San Francisco, and Jean-Baptiste Bord, who is the Attaché for Science and Technology at the French Embassy in the United States. So thank you so much for sponsoring this whole week of wonderful science fairs. I'd also like to thank the QBI events team, Gina Nguyen, Alexa Rocourt, and Carolina Lenzi, who have been working behind the scenes here to do a wonderful job of making everything go well. So thank you so much to all of you. And if anyone has not had a chance to see the science fair and the discussions that went on this week, there is the URL that's available to look at, and they're all on this website. Tuesday's science fair, for example, had a great conversation about the immunotherapy and personalized medicine. On Tuesday, there was this great chat about combining arts and sciences. So they had a cinematographer, an astrophysicist, structural biology. So it was just really a very stimulating conversation. And then yesterday, we had a great science fair on the multidisciplinary team building aspects and how we can collaborate and cross these borders. So today's topic, again, is the global issues on parasitic diseases and collaborations across the board. So borders. And so we will be discussing neglected tropical diseases in particular. And for just a little bit of background, I wanted just to show you this slide. This is the list of neglected tropical diseases, or NTDs. There are about 20 or so conditions or parasitic or diseases here that were designated by the World Health Organization. And these are all diseases that are in the tropical and subtropical areas of the world and affect over about 1 billion people worldwide. So they're called neglected diseases, and we will talk about this in our panel discussion because they really have not received much attention in these research areas of drug discovery and vaccine development, as well as many other research areas. So we will be talking about this over the course of the discussion. Over here on the right panel, you see a number of those diseases, and about half of them are parasitic diseases. So our panel, who will be introducing themselves, have worked on one or more of these NTDs. So if we could start with Gérald, who will be speaking first. Thank you.
I'm unmuting. I didn't forget this. So thank you very much, Judy, and also the organizers for inviting me. It's really an honor to be part of this panel. So my name is Gérald Spaeth. I'm a director of department and head of unit at the Pasteur Institute in Paris. We are working, yes, thank you for the first slide. We're working on this parasitic disease called leishmaniasis, which is a global health threat. Now, this disease comes in different forms and flavors. You see this on the column on the left. We distinguish 3 different forms: cutaneous, mucocutaneous, and visceral leishmaniasis. You can see on the pictures the devastating pathologies that these parasites induce. It is not the parasite that makes these people sick, it's your own immune system. So we call these diseases immunopathologies. On the left side, you see the world map with the countries in red indicating about 98 countries where these diseases are endemic. We have an estimated number of 12 million people infected at any given time, but this is largely underestimated as, of course, this is all happening in very poor countries. Now, the animation, please. If you can add in the animation. We at the Pasteur Institute have the privilege to be part of a very big network of Pasteur Institutes, 33 institutes worldwide. And here I just denote the various Pasteur Institutes in endemic areas with whom I'm collaborating in different projects. Now, we're facing various challenges in Leishmania research, notably, and this is shown in the lower box, we have a current epidemics in different countries. Leishmaniasis is emerging, for example, in North America, Europe, and also Southeast Asia. We have urbanization of the disease, but the biggest problem remains, of course, drug resistance. And all of these challenges are even more aggravated by the facts that we have no vaccine available against Leishmania, the parasite, and the chemotherapy is very limited. Next slide, please. So the infectious agent that causes leishmaniasis is this unicellular parasite called Leishmania. These parasites come in two different flavors or stages. We have a flagellate motile promastigote shown in the lower panel and an intracellular parasite that is not motile, that is residing in immune cells. And this form of the parasite is called amastigotes. Next animation, please. So what characterizes Leishmania, first of all, is that it's adapted to different types of hosts. So you have in the lower part of the middle panel an insect. This is the vector that can transmit Leishmania to different vertebrates. It's not a human parasite, it's a vertebrate parasite, which is, of course, another challenge to combat the disease. Now, these parasites, the different stages, are perfectly well adapted to survive either in the sandfly for the promastigote or the mammalian system, which is the amastigote. Next slide, next animation. The parasites also very much evolve in response to the ecology, and notably the type of insect and the type of animal that is in a given area. And this adaptation or evolution of Leishmania in these different ecologies has quite a big impact on how the disease manifests itself, and so how the parasite is pathologic to the host. Next slide, please. Now, we're investigating this interaction between the ecology and Leishmania and the host consequences as part of a European-funded consortium that I'm coordinating. It's called Leishmaniasis. You see the various collaboration partners. We have experts in high-throughput technologies in the north that collaborate with the experts in clinical Leishmania infection from the south, notably in the Mati countries, which is Morocco, Algeria, Tunisia, and Iran. So I think I want to stop my little introduction here and give the word either back to Judy or the next person that presents.
Great. Thank you so much. So let's see, Chaitan, if you could unmute yourself and then we can hear your introduction.
Thanks, Judy, and thank you for inviting me to speak at this science fair. I'm Chetan Chitnis. I'm the head of the Malaria Parasite Biology and Vaccines Unit at the Institut Pasteur in Paris. And if I can have the first slide. So this shows you the problem of malaria. About 20 years ago, in the year 2000, there were as many as 500 million cases of malaria each year, which led to about 1 million deaths, primarily in children and also pregnant women every year. So that was a huge burden. And since then, we have made progress. That's when the world decided that it was time to try and control malaria aggressively and possibly even eradicate malaria. And you can see the progress that was made since the year 2000 over the next 15 years with a variety of tools, primarily use of insecticide-treated nets, indoor residual spraying, so spraying of houses in endemic areas with insecticides, and early case detection and treatment. The malaria transmission was driven down, and you can see that here in Africa, which is the main place where you have where the malaria problem is the biggest, you can see the malaria transmission going down. Red is the areas with the highest transmission intensity, and that has really shrunk. So we managed to reduce malaria to about 214 million cases and about 500,000 deaths. But since then, if I can have the next slide, we have not been able to make further progress. So we have reached a plateau, and despite the continuous application of these tools, the number of cases and the number of deaths remain static. So what's clear is that we need additional tools to achieve elimination. Ideally, if there's a vaccine, that could be helpful, that could both protect individuals against malaria and also reduce transmission, or new drugs. This is primarily falciparum malaria. The next slide that shows you the incidence of Plasmodium vivax malaria. This is the other Plasmodium species responsible for most of the human malaria. And you'll see that this malaria is actually absent from sub-Saharan Africa. And that's because most of the people in sub-Saharan Africa are Duffy negative. They lack a blood group antigen on their red cell surface, which is used by the parasite for invasion. And that's why you don't find Vivax in sub-Saharan Africa, but it's actually widespread outside Africa. And again, some progress has been made in reducing Vivax, but again, we have reached a plateau. The next slide. So both for Vivax and falciparum, we need to develop vaccines. These could be very essential tools in driving malaria towards elimination. So how do you go about trying to develop vaccines for malaria? So this shows you the different life cycle stages of the parasite, and also shows you how we might intervene at different stages to try and make a vaccine. So for example, when sporozoites are injected into the human host by an infected mosquito, you can try and target the sporozoites with antibodies. The sporozoites go and infect liver cells, hepatocytes, where they multiply and differentiate, and you can try and target these infected hepatocytes using cellular responses. Then the parasite differentiates and is released into the bloodstream in the form of merozoites, which go and infect red blood cells. And you can try and target these merozoites and block their infection of red cells. And finally, some of these parasites in the blood stage transform into gametocytes, which are taken up by the mosquito and are ready to be transmitted again. And there are ways to also target the parasite in the mosquito stages. So number of different groups, including ours, have been trying to develop vaccines targeting these different stages. The next slide shows you the progress we've made in trying to develop a vaccine for Vivax malaria. I mentioned to you that Vivax requires interaction with the Duffy antigen on red cells for invasion, and this is the protein that binds the Duffy antigen. We identified first the part of the Duffy binding protein that binds the Duffy antigen. It's this region 2, this N-terminal cysteine-rich region of the Duffy binding protein. So that's the part of the molecule we have been trying to make a vaccine with. We have solved the structure. We have shown that the binding site is highly conserved, and also shown that upon natural infection, if you make antibodies to this binding site, you are protected against subsequent infections. So we have developed a vaccine that's currently in clinical trials. It's a recombinant protein-based vaccine that is formulated with an adjuvant. It was found to be safe and immunogenic in a phase 1 trial. And we have just completed a phase 2 challenge trial where we have shown that immunizing with this molecule will reduce parasite multiplication rate by as much as 50%. So it's a good beginning. We need to improve on that, possibly by adding another component to this, such as a liver stage component, to try and get synergy and achieve high efficacy. So these are some of the directions we are working in, and I think developing a vaccine could be really a game changer in trying to eliminate malaria. Thank you.
Great. Thank you very much, Chetan. And we will definitely come back and revisit the malaria vaccine for P. falciparum as well in our discussions. So I believe I am next. I would like to introduce the life cycle of Oncotherciasis volvulus. My lab has been working on filarial diseases, and this slide will show you the life cycle, which will also be referred to during the next couple introductions by Fidelis and Kennan. So Onchocerciasis is the name of the disease. It's also called river blindness. The adult worms are found in subcutaneous nodules, shown here on the lower panel. And when you open up the nodules, you can find several male and female worms, and they can be on the order of about 12, 15 inches in length. So they're very large worms. And the female, after she mates, will release these microfilarial stages, or the very early larval stages, into the skin. And these microfilariae will move around in the skin and migrate around the host tissue. And when they migrate up to the eye, there is a very strong immune reaction that occurs to these dead and dying microfilariae, because not all of them will make it and survive. But that's the main cause for the blindness that affects so many people. Then the microfilariae in the skin are taken up by the vector black fly, which, when it takes a blood meal, ingests the microfilariae, and then those larvae undergo a developmental stage in the black fly and to become an infectious larval stage. And then when they bite another person, those larvae become injected into the skin, and they develop into the adult worm. So that's the life cycle. And the main symptoms associated with onchocerciasis is shown here, where the microfilariae in the skin causes very, very severe itching. And then, of course, the main concern is this blindness that occurs. And you can see here a man leading a blinded person by the hand, and often using a stick. And so our goal was, in our project, was to try to identify some new drugs for onchocerciasis therapy. And in the next slide, what you see, and I'm not going to go into a lot of detail on this, but we had to develop a number of in vitro assays to screen our compound libraries. And so again, the goal is to test which one of these drugs could be effective and move on to the next steps. So just to show you, there are a variety of in vitro assays, and Fidelis will also talk about the ones that he used in his assays, the Onchocerca Oshengi. You can see one of his technicians and some of his students worked on this project as well. So I don't want to spend a lot of time on the details of this. I do want to show a movie, though, of these worms. And these are Brugia pahangi, which is a really good, excellent surrogate, because we can't work on some of these worms because, for example, Onchocerca volvulus is only specific to humans. And so we can't work on them specifically, but we have a lot of really good surrogate models. And this movie just shows you, number one, the size of these worms. The wells are about the size of a nickel. And so these worms are about 3 to 5 centimeters. And so we've added compounds to each of these wells. And the first column on the left-hand side shows high concentrations, and then there's serial dilutions all the way down. So that we measure the movements using a computer system, and are able to determine whether some of these compounds are good hits or not to move into in vivo studies. And so from there, I'd like to just turn it over to Fidelis, who will talk about his work and some of the aspects of the work he developed.
With many institutions around the world, including also Judy's lab. We partner with Sarah's lab, Sarah Lou Stigman at the New York Blood Center, and with many pharmaceutical companies around the world, including Merck, Novartis, Eisai. And we have an organization at the level of WIPO, which is called WIPO Research, that is helping to facilitate many of those collaborations for us to develop drugs against the diseases and even vaccines. So we have had quite some challenges because, first, you know that getting a drug for a disease of poverty-related disease like Oncotherciasis, is so difficult in part because people don't want to sponsor. The markets are not incentivizing. So when you produce the drug, the market is not very clear how you're going to make money from that. So because of lack of funding, we have problems getting drugs for neglected tropical diseases like Oncotherciasis. But the parasite is also a little bit more, it has a complex life cycle, very complex. In terms of, that is especially important when you want to develop vaccines. Then that complex life cycle makes it so difficult. The fact also that it's multicellular makes it so difficult to get vaccines against those parasites, like Oncotherciasis. And of course, the limited funding. So these diseases are neglected. They have remained without effective vaccines or effective drugs. And thanks to the collaborations that are ongoing now, it is hoped that in the near future, we will get those drugs and vaccines developed against them. I think that Judy already talked a lot about the disease, how devastating it is, blinding, disfiguring people's skins and making them having a low life expectancy and all of that. So I will come back a little bit later to talk more.
Great. Thank you, Fidelis. And let's turn it over to Kennan, who will then follow up on a lot of this work. Thanks, Kennan.
Thank you, Judy, and thanks very much for the invitation. What I'd like to talk about, by way of introduction, I work for Abbvie in non-clinical development, and we happen to have a compound that's in phase 2 clinical trials for river blindness, which is called flubentylicin. And the question you may ask is, how did we as a large pharma company get involved in this compound particularly? So on the next slide, one of the mechanisms to target for the treatment of Oncotherciasis is Wolbachia. And Wolbachia is a bacteria that's symbiotic in the filarial worm. And you can see this on the slide, how the Wolbachia is inserted into the worms. The Wolbachia is essential for worm development in embryogenesis and survival of the nematode itself. But more importantly, doxycycline as an antibacterial has been shown to be effective in sterilizing the worms and slowing and leading to the slow death of adult worms. The slow death is particularly important given the very large worm burden in each person, as Judy mentioned earlier in her introduction. There's some challenges with doxycycline, however, as the treatment requires at least 4 to 6 weeks of treatment, which is difficult in the African countries. And doxycycline is also contraindicated in children and pregnancy. So how did we get involved with Wolbachia? Abbvie has lots of compounds, and we frequently offer small compound libraries to various groups who are working on screening for new compounds, new mechanisms. And we offered a small library to the Anti-Wolbachia Consortium. Much to our surprise, there was a hit in that small library, and the hit was as potent as the positive control. Further evaluation of that hit, which turned out to be tylosin, showed that tylosin has good in vivo activity when dosed parenterally, but had no oral bioavailability. And so we decided that as a pharma company, we could explore potential modifications to tylosin to improve its oral bioavailability. But then we're left with, okay, we don't have any experience in this therapeutic area at all. Not from a biology point of view, not from an animal model point of view. So we formed a collaboration with the Liverpool School of Tropical Medicine, with the University of Bonn, and with Drugs for Neglected Diseases, DNDi. For the discovery efforts, LSTM and Bonn provided in vitro assay screening for all of our new compounds and helped us with efficacy models in vivo. Abbvie provided the chemistry, the profiling of the compound from an Abbvie pharmacokinetic point of view. We did the tox and did the chemistry scale up, as well as the formulation development. As we moved into clinical development with 4083, which has emerged as a very interesting compound, which is has oral bioavailability and is many orders of magnitude more potent than doxycycline. We ran the phase 1 clinical trials with Abbvie here in the US, but our clinical partner, Drugs for Neglected Disease Initiative, is actually running the clinical trial with us in Democratic Republic of Congo. We're building on their expertise because we have no idea how to conduct clinical trials for this disease. And this phase 2 clinical trial is ongoing now. We are providing the drug in capsules for formulation, and we are also running the bioanalysis for that study. So I'll turn it back to Judy.
Great. Thanks so much, Kennan. Gosh, you know, you made it sound so very straightforward and going, you know, very, very 1, 2, 3, and very quickly. But, you know, this is rare. And something I wanted to talk about with this group is why don't we have more drugs for these neglected diseases? And I'd like to loop back to Kennan and find out how Abbvie decided to, you know, work on onchocerciasis. But, you know, Gérald, maybe you could start off the conversation with why we have so few drugs. Sorry, if you could just unmute yourself, that would be great.
Sorry for the glitch. I think Fidelis already alluded to it. It's kind of a political problem. There has been this London Declaration for Neglected Tropical Diseases in 2012, where many stakeholders came together and decided that they don't want to benefit and profit from the poorest of the poor, and so they waived any kind of intellectual property attached to neglected disease drug discovery. And even though this is, of course, ethically the right way to go, it really took out any kind of incentive for the big pharma to invest in a very expensive drug development process. And so I think that this was definitely a first hit into not having enough compounds or hits and leads that are developed. So for my purpose, from my side, for leishmaniasis is certainly really one of the major limiting factors.
And of course, with the leishmania, the fact that they are intracellular and they multiply within a parasitophorous vacuole makes it so difficult for drugs to get through the system, through the host cell, through the membranes within the cells itself. So this is a very difficult
so we're doing a lot of drug discovery and not development, but just a basic science lab. And so we're using the host cell infected with the parasite to screen directly on the infectious system. So any kind of hit that we get will have the capacity to traverse all the different membranes, of course. Otherwise, it would not work.
Right. Very important. And then, Kennan, can you tell us a little bit about how it came to be that Abbvie worked on onchocerciasis?
So following up on the hit that we had from the initial compound library given to AWOL, one of the things that came out of the Gates Foundation challenging the pharma leaders was that we set up a neglected tropical disease initiative inside of Abbvie, which is based purely on people volunteering to help as experts, as consultants. Maybe we run a few studies. And so we have reached out to several of the non-for-profit groups and offered our services, or they've come back after screening libraries and said, could you help us with X, Y, and Z? It also helped that the head of research at DNDi used to work for Abbvie. And so he knew exactly who did what. And so he could call us and say, would you be interested in helping with this particular project? When we decided to follow up on the Tylosin hit, we had a retired chemist who offered to come back and make analogs. And so he worked a couple days a week and made probably 200 different compounds that we used to screen for improvements in potency, improvements in pharmacokinetic properties, et cetera. And from that came the Abbvie 4083 compound. Once you have a candidate compound, the pharma industry knows well how to develop that compound. So after that, it was relatively easy, but still everybody volunteering to move this project forward.
I think that's amazing. And that's one thing I've really appreciated working on these neglected tropical diseases, is that people really who work on these really have a passion to try to move the whole field forward and get treatments, get good therapies for these terrible, devastating diseases. So it's really remarkable that people are volunteering their time to do this. It's a lot of work. And then maybe Fidelis, can you tell us how you developed some of those collaborations with Merck and the other companies? If we could just unmute.
Okay, thank you. Yeah, so thanks to the initiative, which was recently, well, not recently, it's about 10 years old. It was created at the World Intellectual Property Organization, WIPO. This initiative is called WIPO Research. WIPO, in partnership with BVGH, BioVentures for Global Health, created WIPO Research. This initiative has been soliciting invitations from scientists around the world, and some of us got to know about it, so we applied to join WIPO Research, and thanks to that, they were able to connect us with different pharmaceutical partners and also different laboratories around the world. So we were now able to get those compounds from those pharmaceutical companies who have the compounds but are not interested in developing the drugs. Of course, because like I said a while ago, the markets are not incentivizing enough. When we get those compounds, we then get funding from organizations like Wellcome Trust and the Bill and Melinda Gates Foundation. And of course, collaborating with labs like yours, Judy and Sarah, and we're able then to develop the screens because the parasites are around us. So we developed the test to screen for those, to screen those compounds for those that may be effective against the parasite. And in fact, we have come up with quite a number of them, which we are following up. So we got thousands of compounds from Merck KGaA in Germany, and a handful of compounds also from Eisai in Japan. And then we got training from Novartis in Basel, Switzerland. They help to train in staff development. And those are essentially the places where we've gotten some compounds. But WHO through TDR also had some compounds they sent to us, and some colleagues around the world do send compounds. So WIPO Research has been helping, but also with direct collaboration with laboratories like yours, and with the grants that we got from Bill and Melinda Gates Foundation, we were then able to get compounds from some of the companies that you already you know of.
Right. No, that's great. It's really fantastic that you're able to develop so many collaborations with pharmaceutical companies too, and that they're investing their compounds into this work. Okay. I'd like to kind of turn toward vaccine development and this hot topic of this malaria vaccine. So, Chaitan, would you like to kind of lead the discussion here on the new malaria vaccine,
By that, I suppose you are referring to the GSK vaccine, RTSS, that's been in the news. That's the first vaccine that's been licensed for any parasitic disease. So I think it's a real milestone. Having said that, this is a vaccine for Plasmodium falciparum, and the efficacy of the vaccine is about 39% against clinical falciparum malaria over a period of 2 to 4 years. And against severe disease, the efficacy is around 30 to 32%. So while these numbers may sound low, because you're usually used to hearing efficacy of vaccines to be, at least 80 or 90%, those are considered to be, the goals one would like to achieve. I think for a parasitic disease, even these sort of modest numbers are quite an achievement. And actually, in an endemic area where you have a very high burden and a high risk of mortality from malaria, these numbers, you could be saving, if that protection against severe disease translates into protection against mortality, you could actually maybe, if you save 1/3 of the children from dying from malaria, that is actually a huge benefit. So I think it's an excellent first step, but we need to do better. We need to come up with a better vaccine that provides us a baseline on which to build. Now, this vaccine was based on a single antigen from the sporozoite stage, the antigen is called the circumsporozoite protein. And I think to build a more efficacious vaccine, we might need to add to the circumsporozoite protein with other antigens, either from the pre-erythrocytic stage or from the blood stage. And this is where we really need to invest in upstream, more basic science to understand the parasite, understand immune responses to the parasite, try and identify protective immune responses. And I think with the availability of the whole genome sequence, of being able to analyze human immune responses at a sort of global scale using some of the really new techniques that allow doing things at sort of genome scale, both with the parasite and in humans, I think there are some real opportunities to try and discover the antigens that you might need to combine to achieve vaccines of high efficacy. In addition, the other exciting thing is the availability of some new platforms, such as the mRNA platform, which has been so successful in making the COVID vaccine. Now, this platform could equally well be applied to try and make the next generation malaria vaccine. So I think there are some exciting opportunities in the years ahead. And I think this is where collaborations between academia, between different academic institutions, and with industry could really be very fruitful in trying to achieve some of these goals, such as developing a high efficacy malaria vaccine.
Great. Gérald, would you like to add to that?
In terms of the vaccine development, or
what about for leishmaniasis? What do you think of the prospects?
I think leishmania is a key example, I would say, of how intelligent the smart parasites can be. Because any kind of weapon we have to combat an infectious disease, whether it's control of the infectious cycle or drug discovery or vaccine development, leishmania has its tricks to avoid being controlled or treated or vaccinated against. As I mentioned before, leishmaniasis is an immunopathology, And so I think that we have to do a lot of basic research to understand how leishmania affects the immune system, because vaccines depend on a functioning immune system. So maybe this needs to be really also considered, notably in the leishmania infection, that we're dealing with an immune disease. And so somehow I think, there is a lot of things we don't know yet in order to maybe come up with the vaccine strategy in leishmania.
Yeah, that's a really good point, because I think we have to really consider the host. I mean, these parasites are in intimate dissociation with their host. And so we always have to take into consideration, well, if the parasite dies, what will our immune response do? Will it overreact, or, will it make some other system kind of go awry? So we have to be very careful, again, with these parasites. Because they are these eukaryotic organisms that, after all, they are animals that live within us.
Maybe a question for Chetan, if I may entertain the discussion.
Please.
Why only the protection affects only 30% of the population? Is there anything known whether there is an immunogenetic component to that, or the host immune status plays a role as well? Maybe is it more efficacious in children compared to adults? Is there anything people speculate why the protection is so low?
No, I think people are still trying to analyze that. We still don't understand the immune correlates of protection, so we don't know whether it looks like antibodies may play a role, but it's not everybody with high antibodies that is protected. So it's the immune correlates are not well understood, and so therefore also we don't know whether there's a genetic component to why some are protected and others not. Also, it's not, when you say 30% are protected, it's not that 30% of the children don't get any infections in the subsequent years. It's just the total number of infections in that population goes down by 30%. So a child, instead of getting infected 3 times, may get infected only once, instead of getting clinical disease 3 times over 2 years, may get only one episode of clinical malaria. So it's not very well understood. There's also great diversity of the parasites. So you may be protected against some parasite strains, but not against others. So there's both diversity in terms of the human immune response and genetics, but also on the parasite side.
And Chaitan, you mentioned the new technologies in developing these vaccines. Are there any kind of ideas you have that could be transferred over to your projects, like the mRNA vaccines, for example, you mentioned?
Yeah, the mRNA technology is certainly something that's worth testing. The advantages of the mRNA technology are, since we know now that it's most likely going to take multiple antigens to achieve high efficacy, the mRNA platform is and even to find the right combination, you might need to do test a number of combinations. The mRNA platform makes that very easy, because producing each mRNA construct uses the same technology, and also it's much easier to multiplex and mix and match, and then finally come up with your the best combination. But there are also other technologies, such as the VLP technology has really moved on. That helps to make antigens really immunogenic. What's clear is with antigens in for malaria vaccines that depend on antibodies, you need much higher levels of antibodies than, for example, what you need to get protection against a viral disease. So you need a platform that gives you those very high levels of antibodies. And you may also want to induce cellular responses. And again, the mRNA technology is suited for doing that. So these are the real opportunities going forward in trying new platform technologies to try and develop vaccines with high efficacy for malaria.
Great. So you know, these neglected diseases, they're called neglected because of the lack of attention and resources available to study these diseases. So what kind of advice would you give students interested in working on neglected tropical diseases or parasitic diseases in general, or new investigators that are really interested in trying to help in terms of decreasing the disability and mortality? So maybe, Kennan, do you have any suggestions or ideas? I mean, how did you start out?
How did I start out in neglected diseases? My friend Dale Kemp came and said, I need you to help us with this project. And so I've spent many years in drug discovery and development and thought that I understood that topic until I came face to face with several of the neglected tropical diseases where everything I've learned makes no sense. And so for me, it's a brand new challenge and a totally different way of thinking about things. So it's been very interesting.
Right. And thank you to Dale Kemp for all of his great work. And so Fidelis, what about challenges that you faced being at University of Buea and Bamenda?
Okay, well, thank you, Judy. The University of Bamenda is relatively new compared to, it's 10 years old compared to Buea, which is more than 25 years old. But it is coming up powerfully. Apart from we have this socio-political crisis, which is disturbing the university from taking up fully. However, the university is growing rapidly because new lecturers have been recruited and new buildings are coming up. And then I am in a polytechnic. I'm director of the new polytechnic, which the country is prioritizing. And just recently, we have been given some billions of CFA francs it is being a mark for us to use next year in building laboratories and buying all sort of equipment. And we have biotechnology also on the program. So that is it. We're trying to build a new university and so I have to shuttle, but my lab is still in Buea, the main lab. So I do shuttle between Buea and Bamenda. I think it's fine. Everything is okay. I find it, I don't have much difficulties. I'm still the head of the lab in Buea, the Biotechnology Center. I'm still the head. So no problems.
Great. Chaitan, do you have any advice for some new investigators that want to join the field of neglected parasitic diseases?
For young science students or scientists starting off their careers, I would say these parasites that cause these terrible diseases. First of all, there's some very interesting biology to try and understand. So I think just in terms of the scientific questions that they pose, they are very interesting. They have all kinds of multiple stages. They survive in a mosquito and then also in a human host, and just the way they develop from or transform from one stage to the other, there's some very interesting biology going on. So they're interesting scientific questions. And if you solve some of these key scientific questions, you might actually come up with ways to develop a new drug or develop a vaccine. And that could make a huge impact in terms of protecting people's health and really doing a big service to humankind. So I think it's an area, it's a discipline that combines basic science, basic biology, and translation whether it's drug development, diagnostics, or vaccines, where you can come up with products that are really needed to protect the health of people and of marginalized people who are, who are living in difficult conditions. So I think that's what got me interested in this field. And also on a day, it's a field where you can work in a lab. The next next day you could be working with people in the field. So whether it's in Africa or Asia, so there you can combine field research with basic research and product development. So it allows you to do many different things over the span of your career.
Great. Gérald, any words of advice?
Well, I just have to say that Chetan perfectly well described what I went through. I was attracted to parasitology early on in my studies because I found these parasites so fascinating, how you can have so much intelligence in one individual cell. I tell my students about leishmania, it's the best immunologist that is out there because it knows exactly how to play with your immune system. And so I came through my curiosity and basic science interest into parasitology, worked for a long time, of course, on that level. But when I joined the Pasteur Institute in 2005, I was more and more exposed to my colleagues in the Pasteur International Network. And I must say, I learned a lot from them. So even they might not use the fancy technology, they understand these parasites, they understand the clinics and the disease. And I started to doubt a little bit about all the fancy in vitro systems and experimental systems that I have in my lab. And I really started to change, going away from just purely culture-based parasites, and now we're basically only using parasites coming from the field or being isolated from an animal. And so it helped me to evolve in my research a lot, being of course collaborating with people from endemic areas. And Chetan is right. It's a human experience. It's really a human experience, meeting the people all over the world, working with them, going to field trips, which has it's just a fascinating and fantastic atmosphere, being in the desert in Algeria with the Algerian colleagues, or doing the same in Morocco. So I really can strongly recommend parasitology as a kind of interdisciplinary and international career option.
Great. I'd like to encourage questions from the audience too. We have about 8 minutes left, but here's a question on what are the top priorities for new drugs and diagnostics for NTDs? Is a pipeline poised to address the gaps? What do you all think? Kennan, what do you think? Do you think we is Abbvie, for example, poised to start another project on one of these NTDs?
So I can tell you that we are working very actively with the TBDA in terms of new drugs for tuberculosis. We also are working with MMB on malaria compounds. So we have branched out from Oncotherciasis, but I'm not sure that. So we have picked these based on a lot on the makeup of our compound library, so that we can offer compounds that have the potential to be used as starting points for new discovery efforts.
Yeah, and we do thank Abbvie for continuing on with these programs. I know that some of these pharma companies are moving in different directions and not so much in the area of these neglected diseases. Chaitan, any of your thoughts for new drugs or even diagnostics?
Yeah, so in malaria, are the leading drug, the most effective drug for malaria, artemisinin, which really helped us get out of the problem when chloroquine resistance appeared. Fortunately, artemisinin was there, but now there is resistance to artemisinin, which was first found in Southeast Asia, but now has also been reported from Africa. So there's a real need to come up with new drugs. Fortunately, because of all the work that the MMV, the Medicines for Malaria venture, has done, there is a healthy pipeline of drugs coming out of that pipeline that hopefully should be available. But you need new drugs that will help us tackle artemisinin resistance. In terms of vaccines, we've talked about that. I think vaccines could be a very important tool to help us drive malaria to elimination. It's actually probably even more important for Vivax because of the unique biology of Vivax. It's actually more difficult to control than falciparum. And you need better diagnostics. For example, in Vivax, one of the key problems is the ability of Vivax sporozoites to form latent stages in the liver called hypnozoites. So not all the liver stages come out in the blood. Some can stay dormant for months or even years. And we have currently no way of detecting an individual with hypnozoites. And so you could clear the blood stage infections, but clearing hypnozoites, the drugs that we have some limitations in terms of safety. So we need new drugs. First of all, you need to be able to identify people with hypnozoites. So you need really new tools, new biomarkers that can identify people with hypnozoites, and then you need new drugs to treat hypnozoites. So there is work going on. People are trying to identify biomarkers in terms of immune signatures, antibody signatures that might identify people who have been recently infected and therefore might have hypnozoites. And there is also work in trying to develop new drugs against hypnozoites. So there is work going on, but these are the gaps that will need to be filled to be able to achieve elimination of malaria.
Well, I think it's really important that we don't silo ourselves into just saying, oh, well, we'll find a drug without knowing what that drug is doing in the field, like the diagnostics aspects of it. How effective are these really on people that are actually infected with the disease? So it all goes hand in hand. You have to do vector control, the new drugs, the diagnostics, the vaccines. So as you mentioned, there's lots to do, and even the basic biology of many of these parasites is really critical to moving this whole field forward. So very, very good. We have about 3 more minutes left to our discussion. Would anybody like to add anything else at this point?
Yeah,
again,
maybe
words of wisdom.
Yes, just to follow up on what Chetan was saying, and of course overcome some limitations in the drug discovery program in Leishmania. We're working a lot on Leishmania evolution, and I told in my introduction that parasite Leishmania is evolving very fast towards a drug-resistant phenotype. And so we in the lab, our top priority, based on this finding, is now to develop host-directed drugs. So Leishmania is an intracellular parasite, and just like a virus, it's really plugged into the metabolism of its host cell. And it's really subverting the entire functions of the macrophage. And if we can pull the plug by treating the macrophage, so we're not treating the parasite, because that would drive the evolution of drug resistance, we try to make the macrophage remember, the host cell remember, that it's actually a very strong immune cell. And I think this is maybe, for me, one of the highest priorities. I'm engaged with the DNDi also on this, I would say, novel treatment option in parasitic diseases. It's already, of course, used for viral diseases and also some bacteria. So I think this is maybe something that can respond to this big problem of drug resistance.
Great. Let's see, I think we have one question. Oh, yes, go ahead, Fidelis, please.
Okay, so the top priority for for drugs in onchocerciasis now is the development of an adult worm drug, a macrophylaricide. And to get that done, we will need a lot of philanthropy. Of course, since the markets are not so incentivizing, we will need quite some input from philanthropic organizations like the Gates Foundation, Wellcome Trust, etc. That's what I wanted to say in the case of onchocerciasis. Thank you.
Great. Thank you so much. Well, it looks like we have run out of time. I don't know if we can even address this last one. I know that we do try to engage the public, and we try to get ourselves out there by teaching as much as we can, by doing these seminars and meetings. So thank you all for joining today. I appreciate the wonderful work that the panel is doing. So thank you all, and we'll see you. Stay safe. Bye-bye.
Thank you very much. Thank you. Bye. Bye.